Hello everyone, welcome back to another lesson. Today we're going to be talking about the methods of microbial enumeration, so just the different ways that we are able to count and quantify the amount of microorganisms on different surfaces or products. So we have a few questions from the last lesson. I just want you to think about these. You don't have to turn it in, but do consider all of these different questions. Just think about them, and then just on your own time, just maybe come up with an answer to it. Might be on the quiz. So, why perform microbial counts on our food? So we are going to be using this as an index of hygienic conditions. So just going to tell us how clean the different surfaces that the food came in contact with during the processing, then making sure that the shelf life of the food is going to be good just by determining how many of these food spoilage organisms are inside. And then we also want to be able to detect any possible health hazards or pathogenic organisms that are in our food, because that'll be causing a lot of issues. A few of those are Salmonella and E. coli. So we have a few different methods of enumeration for viable cells. So we have our plate count method, most probable number, and membrane filtration. We'll be talking about all those today. So why would we only want to count the viable microbes in food? So this is because the viable cells are going to only be the ones that are going to be causing us any issues. So all these issues that we just talked about in the previous slide. So hygienic conditions, shelf life, health hazards. So if you have dead cells, then they aren't going to be causing food spoilage or any pathogenesis, anything like that for the most part. So they also aren't going to be able to reproduce, so they aren't going to be able to cause the food to spoil even more. So we're really only concerned about the viable microbes. So the first method is the plate count method. This is going to be the most accurate and widely used method for viable counting, and the most common method of this is the aerobic plate count. So that's going to be known as APC. So this is going to indicate the level of aerobic microorganisms in a product. So some people are going to also refer to this as a standard plate count or the total plate count. So these are all the same thing. So again, not all microbes are going to be counted. So it's all dependent on the medium and growth conditions. So depending on the temperature and everything that you're going to be using, and then all the different nutrients and carbon sources in the medium are going to decide what organisms can grow. And then also, since this is going to be aerobic plate count, then only organisms that can survive in the aerobic conditions will be able to be measured or counted. So, which one of these would not grow? Aerobic, facultative, or anaerobic? And so, aerobic is going to be fine. Facultative can survive in aerobic or anaerobic conditions. And then these anaerobic organisms are not going to be able to survive because the oxygen is present. So our plate count method, the first step is going to be to prepare 10-fold or 100-fold cereal dilutions. And then we're going to be, for this one, you'd be plating in duplicate. So you get two different plates at each dilution. And then that way you have kind of a backup to see how it's going. You take the average of those two. And then after you incubate, you're going to count the colony forming units, okay, CFUs. So the most commonly used amount is 30 to 300 per plate, but for the FDA's BAM, we are going to be only looking at 25 to 250 colonies per plate, okay? So since this is food microbiology, we'll just be doing the FDA BAM methodology as opposed to the most commonly used for other microbiology classes, so be aware of that. The amount of colonies is a little bit different per plate when working with food products like that, okay? So for a cereal dilution, here's an example of how it works. We start with our dilutant in here, or 9 milliliters of liquid in here. Then we have 1 milliliter of our original inoculum, and then that's going to create a 1 to 10 dilution there. So now we have a tenth as many organisms as in the original inoculum per milliliter. Then we do this again. Now we get a 1 to 100. You do the same thing again. Get a 1 to 1,000. and you place another 1 milliliter and do 9 milliliters here, and you get 1 to 10,000. You can keep going until you get countable amount of colonies, okay? So when it's within the range of 25 to 250 for BAM. Now, we have a few different plating methods for plate counts. So they're all pretty much going to be using these serial dilutions, unless you're taking directly from your inoculum. But they're going to have slightly different procedures for how we're going to perform the test and then record the results. So, for the pour plate method, this one is going to involve placing your inoculum into the liquid medium. Okay, so you're pouring in the liquid medium, that's why it's called a pour plate. So you can take either 1 milliliter or 0.1 milliliter for this, and then once you put the inoculum into the plate and then you put the melted agar on top, then you will swish it around a little bit, and that way the agar and the medium with your organism is evenly distributed on your plate. And so keep in mind that not all the colonies are going to be on the surface. That's because they're going to be contained within the agar, a lot of them, since you are placing them not just on the surface, you're placing them within as well. So keep that in mind. So on this PowerPoint, you'll see that there's videos, so you can either use the link right here, or you can just use the link that's on Blackboard. They're all going to take you to the same place, okay? Okay, so now the next method, known as a surface/spread plate method, is going to involve taking, doing your serial dilution again, but then instead of mixing your inoculum with your medium, you're going to have the solidified medium already ready, and then you place whatever sample you're looking at on top and spread it around. So instead of pouring it all together, you're spreading it onto the surface, okay? So you're spreading your whatever liquid you're testing, and you're just going to be spreading it onto the top of your surface, okay? So now all the colonies are going to be on the surface, and the size should be pretty uniform in comparison. So the next one is going to be Petri film. So this is very similar to the surface spread plate, but it's already so it has a pre-made medium, and you're not going to be pouring anything. But this is going to be more industrial scale, so it's a smaller product, takes up less space, so it allows you to save space in the incubator, and it's also designed just to be a little bit easier to use, a little more user-friendly, but it's also going to be more expensive. So with this, you just take 1 milliliter of the liquid, you place it on, and then you're going to be able to count how many organisms you have, either based on the square centimeter or the total liquid amount, okay? So there's a video on Blackboard for that as well. So for the most probable number sets, these are going to be another form of enumeration. So unfortunately, we're not going to be working with any of these because this lab's online, but an example of this is the SimPlate, which is going to be counting based off of a statistic estimation. So these are, again, going to be a commercial plate, kind of like the Petri film. So they're going to contain these substrates that will produce either a red or pink color in these wells. And so the number of red wells correspond to the total number of microbes, the predicted statistical amount. So it's just estimating how many are going to be in that sample just based off of the amount of chemical reactions that take place that change your wells into a pink color. So you can just record how many there are and then compare that to a table to see the estimated amount. So the next method we have is membrane filtration. So this is going to be commonly used to count coliforms in water. So this is specifically for liquids, particularly water. So how it works is you have a little sheet here, cellulose membrane, with a very small pore size, and that's going to allow it to trap any microbes that pass through. So the microbes will not be able to pass through, but as the water passes through, the microbes will be trapped onto the surface of that cellulose membrane. And then once you incubate that, you'll be able to see. And because of the different substrates inside of that cellulose membrane, you'll be able to see the coliform, and they'll have a characteristic metallic sheen. Okay, so this is specifically going to be for water, and that's up to about 100 milliliters for that method, okay? So for our aerobic plate count, APC by BAM, as I was saying, so you're going to be doing your dilution, and then from there, you'd be creating duplicate plates. So you have a 10 to the negative 2, 10 to the negative 3, 10 to the negative 4. You're going to have 2 of each one of those dilutions. Okay, so just make sure you understand this concept. Unfortunately, we're not going to be doing that. So for part B, this is a surface plating method. Okay, so again, you can see the video here, or you can just check it out on Blackboard. And so make sure you're always going to be doing 0.1 for the surface plating. Okay, so it's different for pour plate. You'll do 1 or maybe 0.1, but for surface plating, it's very important that you only do 0.1. Because if you do 1 milliliter, there's going to be too much liquid for the surface plating method, and the top of your plate's just going to have too much. It's not going to be nice. Okay, so then Petri film. This is the last one, so make sure to watch the Petri film demo video to get an understanding of how this one is used, kind of the idea of the applications, why you would want to use this over just, say, a normal spread plate. And then sim plate. So again, this is our statistical estimation method. And so make sure you take a look at this procedure. You're not going to be expected to memorize it or really do anything with it, but I'd like you to be able to take a look and kind of see what you are missing out on. So if you have any questions about this, feel free to let me know. Again, there's going to be your quiz and discussion board post on Blackboard, so make sure to complete everything. And with that, that's it. Good luck, everyone. Have a good week.